Welcome back, everyone, and we are going to pick up where we left off in our last part of our Chapter 12 PowerPoint. So that is 12.3. Here are our learning outcomes for this section. We're going to talk about the four major categories of our second line of defense. We'll talk about phagocytosis and the steps of it, as well as the steps of inflammation. These are both things that we've mentioned before, but we haven't discussed the details yet, so we'll get to that now. We'll discuss the mechanisms of fever and how it develops and how it helps to defend the body. We'll talk about the different types of antimicrobial host-derived products, and we'll also compose an overview about the purpose and the mode of action of complement. And complement is basically a collaboration of various proteins and chemicals in the body that aid in our defense system. So let's move to our first slide here of this section, our second line of defense. We finished with the first line of defense. So now the second line of defense is still nonspecific. Remember, both the first and second lines of defense are nonspecific and generalized. They are part of the innate immune response, and they are going to support our immune system with non-specific responses. And sometimes they'll interact, though, with specific immune responses, and we'll get to that later. But here are the four categories of things that can happen during the second line of defense, one of which we've mentioned before. We have our phagocytosis, which is cell eating. We also are going to talk about inflammation. We will talk about fever and those antimicrobial proteins. So we'll start with phagocytosis. So the general activities of phagocytes is to provide surveillance to various tissues, find microbes that are not supposed to be there, and also to eliminate them. Now, that does not just include, however, pathogens, but also injured and dead cells. They can sequester those as well. And also, they will read any kind of immunogenic information, antigens, those markers that we talked about that exist on pathogens, to mark them for destruction and to destroy them. There are three types of phagocytes. We have talked about two of them, are monocytes and macrophages. The monocytes are basically macrophages that have not exited the bloodstream yet, and our neutrophils are a separate type and are the most common. So these are examples of phagocytes, are cells that eat stuff. Neutrophils are a general purpose phagocyte. They react very early in the inflammatory response to various foreign substances or to damaged tissue. We have more of them in our body than other cell types, but if we have an even higher than normal neutrophil count in the blood, that is a really common sign of a bacterial infection. So, for example, you people that are going to nursing, you may be familiar with a complete blood cell count, or they'll shorten it and say a CBC one day. And that CBC typically is going to have lots of information on it, but part of that is a white cell count and even a differential, which shows us what percentage are neutrophils, what percentage are macrophages, what percentage are lymphocytes, B cells, T cells, and so on. So if we see a higher number than usual of white blood cells, and particularly of neutrophils, it's typically indicative of a bacterial infection. And these neutrophils are the primary component of pus whenever we have some kind of bacterial infection. Monocytes. These are, again, transformed into macrophages when they migrate out of the bloodstream. And at that point, they will leave the bloodstream and go to various tissues. So at this point, the macrophage can have different names depending on location, which I know is kind of a pain in the butt. But monocytes are in the bloodstream. When they leave the bloodstream, we call them macrophages. And then when they get to where they're going, we call them something else. So, for example, we can have alveolar macrophages. And those are found in the lungs. We have our Kupffer cells. These are macrophages in the liver. Dendritic cells, are macrophages in the skin. There's macrophages that can be spread throughout other places too, like the spleen, lymph nodes, bone marrow, kidneys, the bone itself, and the brain. But we don't need to necessarily know any further naming systems. And even further than that, there are macrophages that do just kind of drift around the MPS, which is that mononuclear phagocytic system, this stream that exists throughout the internal and external, or excuse me, throughout the external fluid throughout the body in between cells. So again, we said these phagocytes are cells that eat. They perform phagocytosis, which is the physical process of engulfing some kind of foreign material and dismantling it. It can be an isolated event or as part of an orchestrated, bigger part of an event of inflammation. But ultimately, the steps that exist in phagocytosis are as follows: chemotaxis, ingestion, phagolysosome formation, destruction, excretion. Let's go through each one of those. So here, this is a table from your book that gives some descriptions of each. However, I like to look at pictures when I lecture, so because I don't like the wordiness of this. I don't expect you to memorize this by verbatim either, but you should know this order, know the order, and a little bit about the what happens. So what we're seeing first is chemotaxis by a phagocyte. What is chemotaxis? Chemotaxis. Think of it this way. Chemo means chemical. Taxi, what does a taxi do? It takes you somewhere. So chemotaxis of a phagocyte is where we have attraction of this phagocytic cell to a foreign invader or to a site of damage. Like if I step on that rusty nail, that's going to attract. There's going to be chemicals that are going to release in my damaged cells that are going to attract phagocytes. So that's what chemotaxis is. It is the attraction of phagocytic cells to a particular site of damage or infection. Number 2, we'll have adhesion of bacteria. Number 3, we'll have engulfment of this particle. So again, we see here we have our example of our bacteria. And as a reminder, don't forget, bacteria have those PAMPs. Remember, those are those collection of markers or antigens that most microbes have, like bacteria, and they're going to be detected by our PRRs. Those are those recognition sites that are found on our white blood cell. So whenever they feel around and feel those PAMPs, they're going to engulf that bacteria. And so that's step 3. We can see them engulfing the bacteria. So this bacteria has been engulfed and moved inside of the cell. So now what we call this is the phagosome. It is a vesicle or a vacuole that contains our bacteria. Now, something you see on this side are some lysosomes. And so when y'all talked about cell structure with either me or in your whatever biology class you took or A&P, hopefully y'all talked about lysosomes. And they contain enzymes like lysozyme. And when I teach this in biology, I say think of it like Lysol. Lysol cleans, right? So these lysosomes are filled with these lysozyme enzymes, and it's going to fuse, as you can see, with that phagosome that contains the bacteria. And at that point, we call it a phagolysosome because the phagosome has fused with the lysosome. And so at that point, we now see the lysozyme enzymes that are infiltrating and able to come in contact with these bacteria. That lysozyme, again, we said is like Lysol. It's going to kill and destroy these bacterial cells, and then the residual debris will be excreted from the cell. All right, so again, that's the same thing we saw in this picture. Chemotaxis, we got to have our phagocytes are going to move to an area where there is some kind of damaged cells or infection, the phagocytes are going to use their receptor sites, the PRRs, to recognize the PAMPs that are on bacteria cells and cause engulfment. That will form the phagosome. Then our lysosomes will fuse with the phagosome to form the phagolysosome, and that will result in destruction of the bacteria and then finally elimination of any debris. So that's the extent of what you should know for the exam. You don't need to know all of this verbatim. Okay, so what are examples of PAMPs? We've talked about them, I know, a couple times at this point, but let's go into a little bit more details. So we have signaling molecules that are found on microbe surfaces that are recognized by our phagocytes or any other defensive cells. And again, we've said that these molecules can be shared by many different organisms, bacteria, even eukaryotic pathogens like protists may have them, but these are combinations of proteins, antigens, or markers that are not found on mammals, which is why our immune system detects them as red flags. So that collection of proteins, our body knows. We don't see that on, I mean, it doesn't know that we don't see it on mammals. We know that as humans because we've studied this, but our immune system has learned that we don't see this typically in our own cells. So remember, it's about recognizing self versus non-self. So this PAMP, this collection of antigens or markers that we see in these microbes commonly are red flags for these phagocytes and initiate this process of phagocytosis. So some common bacterial PAMPs would be peptidoglycan. So recall when we talked about eukaryotic cells, animal cells don't even have a cell wall. They definitely don't have peptidoglycan. Eukaryotic cells that do have cell walls, like plant cells, are typically going to be made up of cellulose or chitin for fungi, for example. And there's some other examples in different organisms. Another one in plants would be lignin. But the point is, we don't see peptidoglycan, which again is a protein sugar complex, in cells other than bacteria cells. So that's why it is referred to as a PAMP. It is a pathogen-associated molecular pattern of proteins and sugars that we don't see on cells besides, in this case, bacteria. Another example that we've talked about before was our lipopolysaccharide. That specifically is a lipid and a sugar compound that we only see in gram-negative bacteria. So that combination is also serving as a marker that is a red flag to our immune system. Viruses have PAMPs, which include double-stranded RNA. Can you guess why? Do we have double-stranded RNA in a eukaryotic cell, such as our cells? No. So something, again, you should have learned in A&P or biology is that DNA is double-stranded, but RNA is single-stranded. Viruses are weird, though. Recall our chapter 5. We said they have weird things that they do and weird structures sometimes. And they do have, in some cases, double-stranded RNA. So because we do not have that as humans or mammals in general, that is a PAMP, a pattern in this virus that would be a red flag for our immune system as humans. All right, so what about in the host, the receptors? Again, these are called PRRs, and they're found, of course, as we've talked about, on phagocytes, which can include dendritic cells. They're sometimes found on epithelial cells and even on lymphocytes. But again, they bind and recognize these PAMPs, and they are going to have those recognition sites regardless of whether or not they've come in contact with a PAMP before. And that's exactly why, once again, we refer to it as the innate immune system. It does not have to have seen these things before to know that it's a red flag. Another thing that exists are inflammasomes. These are receptors that are found in the cytoplasm of phagocytic cells. Okay, let's pause that for a second. Let's discuss the difference here. The PRRs, Remember, those are in the cell membrane. Lots of proteins and sugar protein complexes that can exist within the cell membrane. The inflammasomes, on the other hand, are found within the cytoplasm inside of the phagocytic cells that can initiate an immune response. When they recognize any PAMP that has been phagocytized, it will lead to the release of chemicals that will initiate some other response to help regulate inflammation, hence why they're called inflammasomes. All right. So our next description we are going to discuss is how we identify an inflammatory response. And there are a couple of classical signs and symptoms that are required for us to identify inflammation. The first one is rubor. Rubor means redness. And that redness is caused by an increased amount of circulation of the blood in that area due to vasodilation, meaning that your blood vessels are going to get wider, so more blood comes to the area at the site of an injury, so it can bring on more immune system cells with it. Also, we have calor. This means warmth, and that is the heat given off by that increase in blood flow. We will have a tumor, which is swelling that is caused by the fluid. I feel like another more common word for this is edema. We have dolor, which is pain. And then we finally can have loss of function in the case of an inflammatory reaction. Alright, so some chronic diseases like cardiovascular disease are caused by this chronic inflammation. It can be local, it can be systemic. Aging is a consequence of increased inflammation in multiple body systems, is what some researchers believe. So if we can control inflammation, maybe potentially we can slow down the aging process, something to think about. Factors that can elicit inflammation includes trauma from infection, because infection causes damage to tissues. Tissue damage due to just blunt force injury or necrosis due to chemical agents or physical things, and even adaptive immune reactions can cause inflammation. So what are the functions of inflammation? And we kind of think of it as a bad thing almost initially, because it's not fun. We see, if we feel inflammation, something that feels swollen, it hurts, it's warm, and it's not a comfortable feeling. But despite that, inflammation does have some good functions. It helps to mobilize and attract immune cells. So again, we're having an increased blood flow to the area, so more blood cells, including white blood cells, are going to go there. It's going to help to set into motion various mechanisms to repair that damaged tissue. Oh my goodness, you guys, I about jumped out of my chair. I don't know if y'all heard that, but I have somebody in the room next to me with a drill. Oh my goodness. Alright, you guys, let's start this slide over because I literally about came up out of my chair. They were drilling into the wall right next to the wall next to me, and I almost had a heart attack. So, back to inflammatory responses. So, like we said, some of these chronic diseases, like cardiovascular disease, are caused by chronic inflammation. So, this can be local systemic. We also believe that, or at least some researchers believe, that aging is a consequence of inflammation over a long period of time throughout our life. Interesting thing again. So can we control inflammation? Can it extend life? And some factors that elicit inflammation, like we mentioned, is trauma from infection, tissue injury, which is another type of trauma, both of which can lead to necrosis due to physical factors or chemical agents that can cause cell death, and even adaptive immune reactions themselves can cause inflammation. So next, what is the function of inflammation? So, like we said, we feel, initially like it's not a very great thing, but it does have some good things that come along with it. So, we have one of the functions of inflammation is to mobilize and attract immune components. So, again, that plays into our increase in blood flow to an area. It will attract more platelets, more immune system cells, both of which are going to play a part in either healing or immune reactions. It can aid in setting into motion repair mechanisms. It can aid in the destruction of microbes, block further spread of an infection. That inflammatory response is really powerful defensive reaction, even though it may be uncomfortable for us. But it's a mean for the body to maintain stability and restore itself to have time to heal after injury. And it has the potential to cause tissue injury itself, destruction, and disease. So it's got some pros and cons. So as long as it's maintained and under control, it's actually a good thing. It's when it gets out of control that it can lead to negative impacts. What are the stages of inflammation? So the stages of inflammation are going to lead up to a process that is dynamic, it's predictable, it's a series of events or a cascade of events that can last for variable amounts of time. But initially, there's going to be some kind of origin of inflammation, typically an injury, that will cause a chain of reactions to occur. I like to call this a cascade of events that will then lead up to this inflammation of the injured area. So here we have our stages of inflammation. And again, as per usual with these big tables, you do not need to memorize all of this verbatim. Know the list here in order and a little bit about what they mean. So we have injury that happens first, which is when our immediate reactions happen. We have vascular reactions, edema and pus formation, and resolution / scar formation. So let's look here at some pictures. Here what we're seeing is our initial injury. We see this nail that is going into the skin right here. And this nail, or whatever it is, needle, I don't know, it doesn't say, it's probably a nail, but it has bacteria. And so now we have created a wound that is going to have bacteria in it. So over time, what will happen is this wound will heal. We will clot our blood, prevent it from flowing out of the body, and repair the blood vessels as well as the damaged skin tissue. And we will take care of any kind of bacteria cells that have infiltrated into our skin by utilizing various white blood cells. For example, these just happen to be neutrophils. I can tell that by the way the nucleus looks. I wouldn't expect you to know that, but you should know that it's an immune system cell, and you should know that it's most likely a phagocytotic cell, or a cell that does phagocytosis. It consumes the bacteria. All right, so looking at our next images, we are seeing a few different things here. We're seeing in the red areas, notice around here, rubor. Remember, that's redness. In this area here, we see edema. Why do we see edema? Again, we have broken blood vessels here. All right, so we're going to have an increase in blood flow to this area, and that's going to cause that swelling or tumor. And I prefer to use the word edema because tumor kind of gets confusing. So then after a while, once these blood cells, these white blood cells have done their job, you'll see pus formation as a byproduct of this process, which also furthers that fluid collection, aka edema. But we will eventually see the tissue becoming healed, and that's the stuff in green. We repair the blood vessels. We have a scar tissue that is seen here. We have in the green cells our lymphocytes that have now come in to assist in our immune response and inflammation. We also see some macrophages that are helping to take care of more of these bacterial cells. So how do they get there? There's two mechanisms that we'll discuss. One we've already talked about, chemotaxis. That's the migration of cells to a stimuli. So again, remember we said that when tissue gets damaged, it can release certain chemicals that will attract white blood cells. So it's almost like, remember, a chemotaxi. It's a taxi that takes our white blood cells to the site of a chemical that is being excreted by a damaged cell. So our cells are going to literally almost swarm from various parts of the body to this site of injury. Now, another term to be familiar with is diabetes, not to be confused with diabetes, not the same thing by any means. So diabetes is when these white blood cells migrate out of blood vessels into tissues. Remember, we talked about that, how our cells, one of the cell types inside of blood that is a white blood cell was called monocytes, and we said that they leave the blood and they go on to other areas of the body, and that's when we call them macrophages. The process that they do to leave the blood and to go to a tissue is called diabetes. It's literally moving these white blood cells out of the blood and squeezing them out of the blood vessels into a tissue. All right, let's look at a picture. So here what we're seeing is a blood vessel. You can see it's made up of a single layer of cells that we call endothelial cells. And this we see are the white blood cells, and you can see them literally squeezing themselves in between the spaces that exist between blood vessels. It's forcing themselves out. And again, that's due to chemotaxis. So there is some kind of chemical somewhere in the body that is attracting these white blood cells, and they're literally squeezing themselves out of the blood vessels and into this extracellular matrix or this tissue where we have some kind of chemotaxic factor, where we have a site of injury. Remember, chemotaxis is going to release chemicals due to infection or site of injuries. So that's what the difference is. Chemotaxis is what's attracting these white blood cells. Diabetes is the blood cells that we see in the image here are white blood cells. These specifically are neutrophils that will leave the blood by squeezing out of the blood vessels and go towards our chemotactic factors. And so you can actually see a real picture of this. It's the transmission electron microscope. Here we have our blood vessel, and these things here, and this would be the cytoplasm in white of our neutrophils, and then this would be the nucleus. And so you can see them like squeezing out of this blood vessel. Now, what are the benefits of these blood vessels allowing this to happen? So, our blood vessels, because they're made up of these little tiny thin sheets of cells, are pretty flexible, and they're, in a way leaky, because they allow for this influx and outflux of white blood cells as well as other fluids and stuff. So that includes dilute toxic substances even. So even though that can be seen as like a drawback in some situations, it has more of a benefit to the body than cons. So this allows for fibrin clots to trap microbes. This prevents further spread, and so the fibrin clot is a step in the process that occurs whenever you have an injury such as the nail in the foot example. There's going to be a cascade of events that happens called blood clotting, and I can't remember if we're going to go into that in this class. I know y'all have seen it in A&P, but basically it's a cascade of events that involves a lot of different blood clotting factors that ends with a fibrin clot that almost forms like this meshwork of a patch that will prevent blood loss from a blood vessel and thus prevent further spread of infection, because these microbes can get entrapped in that little clot. Neutrophils, they also aggregate at the site of infection or injury, and they will be involved in phagocytosis and consuming and destroying any kind of debris, which includes bacteria or whatever is causing and could cause an infection, any dead tissues from trauma like the damaged cells due to stepping on this rusty nail, or any other kind of debris that needs to leave. Now, as a side effect of this process, it will yield pus. And pus, I know y'all have seen that. Y'all know it's that really nasty looking white gooey stuff that comes out of, a great example would be like pimples, but they can, when it comes to infections, they don't necessarily have to initiate as a pimple. But any hoodle, what that is, it's a mass of cells like bacteria, white blood cells, and lots of cell debris. Is all that is, and it causes that that swelling in the local area. The bacteria that are causing the infection or this pus formation are called pyogenic bacteria, pyo meaning pus, genic meaning creation of, and bacteria that are pyogenic are things such as streptococci, staphylococci bacteria, gonococci bacteria, meningogonococci bacteria, and all of those are pus stimulating bacteria. Next, let's move on to fever. So, fever is an abnormally elevated body temperature. That's probably a universally known thing. And it's a universally known symptom to infections in general. But it's also associated with other things like allergies. It can cause fever. Cancers can as well. Other organic illnesses can also cause fevers. But if the cause is unknown, we call it a fever of unknown origin, or a FUO, not to be confused with UFO, unidentified flying object. Haha. Okay. So our body temperature is going to be typically maintained around 98.6 degrees Fahrenheit. Get used to seeing Celsius, you guys, because it's going to be more and more common as you get into your sciencey fields. 97 degrees Celsius is body temperature in our Celsius scale, which is our universal scale for measuring temperature across the scientific field universally. And this is controlled by the hypothalamus, which is a component of the brain. Low-grade fevers are anywhere from 37.7 to 38.3 degrees Celsius. That translates into 100 to 101 degrees Fahrenheit. And getting higher, to a high-grade fever, we're talking 104 to 106 degrees Fahrenheit, which translates to 40 to 41.4 degrees Celsius. Things that cause fever are called pyrogens. Pyrogens. Now, before we go on, I want to backtrack for a second. Pyrogen, as you see here, let's back up. Pyogen. Y'all see the difference? The R is missing here. Pyogenic bacteria cause pus production. Pyrogenic bacteria cause fever. Can bacteria be both? Yes. Alright, so pyrogens are any substances that are going to cause our thermostat that is set by our hypothalamus to deviate from what is normal, aka cause fever. Those pyrogens can be exogenous or endogenous. Exogenous pyrogens are products of infectious agents. These are things such as the infectious agents are things like viruses, bacteria, protists, fungi, endotoxins that can be produced by bacteria, blood products, if you're given the wrong blood product. Vaccines can cause fever. Any kind of injectable solutions coming from outside the body in general, and vaccine would be an example. Those are all pyrogens. They can initiate fever. Endogenous pyrogens, these are pyrogens or chemicals that cause fever that are liberated by monocytes, neutrophils, or macrophages during phagocytosis. So the difference being here if you think about these terms, exo versus endo, exo means outside, exit, endo meaning inside. So the exogenous pyrogens are chemicals that are causing a fever that come from outside of the body. Whereas endogenous pyrogens have been consumed inside of monocytes or any other kind of phagocytic cell. And whenever it consumes and lyses and destroys those cells, it releases some kind of pyrogen that was inside that infectious cell, such as a bacteria. And those could be things like interleukin 1 or tumor necrosis factors that would then result in fever. So again, exogenous means it comes from outside the body. We get this external source inside of us, and it itself, it acts as a pyrogen. And then endogenous pyrogens are when our body engulfs and destroys something that is holding a pyrogen inside of it, like interleukin 1 or this TNF, tumor necrosis factor, and it's liberated by that process of phagocytosis. Now, fever's a pain in our heads, typically, right? And that seems like a bad thing, but just like before we talked about inflammation, it has some benefits to it. So fever is actually a mechanism of inhibiting multiplication of some microorganisms, including viruses, some of them anyway, some fungal pathogens, and even some bacteria. So it's actually beneficial for us to have a moderate to low grade fever to inhibit spread of further infection. It also interferes with the nutrition of many bacteria because it reduces the availability of iron in the area, and thus inhibiting bacterial growth, because many bacteria rely on iron in order for their metabolisms to function. It also increases the metabolism of our own cells and stimulates our immune reactions to happen quicker. So it speeds up hematopoiesis, which is again the production of more blood cells. It speeds up phagocytosis, and it speeds up other immune reactions. So all in all, there's several benefits of fever, even though it can kind of be a damper on our parade. How do we treat fever? Well, it can be difficult to decide whether or not to treat it or not. A slight to moderate fever in an otherwise healthy person should be allowed to run its course technically, because again, we said it has some good things about it. There are some side effects of fever. It can cause tachycardia and tachypnea, which is a rapid heart rate and elevated respiratory, I'm tripping over my words, rate respectively. It can also lower the seizure threshold, particularly in children. However, if you have a high fever or a prolonged fever, and you have some kind of comorbidity, such as like cardiovascular disease, any kind of head trauma, a seizure condition, respiratory conditions, then we would think, okay, we might want to treat that more immediately. But again, if you're otherwise healthy and you got a low grade, maybe moderate, you might just want to let it do its thing. All right, so our next item of concern in our discussion is antimicrobial products. So just as a reminder, I'm going to move back for just a moment. This is a slide much earlier in the slideshow. This is slide 39, but when we started talking about our second line of defense, we said we're going to talk about these things: phagocytosis, inflammation, fever, and antimicrobial proteins. So now we're at the end here. We've talked about the steps of phagocytosis. We've talked about inflammation and what it is, and its mechanisms, and how it's good and how it has cons. We talked about fever, and now we're moving into this antimicrobial protein discussion. So antimicrobial proteins, there's a lot of them, and we're going to talk about some of them. One of which we'll talk about is interferon. I don't know why, but this is probably one of my favorite ones. But an interferon is a protein produced by certain white blood cells or even tissue cells Originally, they were thought to be directed against viruses, but however, it has now been known to be involved in defense against other microbes and in just immune regulation in general. There's different categories of interferons like alpha and beta and gamma, and that's going to be dependent on the source that they are produced by. So, for example, interferon alpha and beta are produced by lymphocytes and macrophages and even fibroblasts, whereas gamma interferons are produced by T cells. But ultimately, what these do is that they can have actually a couple of different mechanisms, but ultimately have the same effect, which is prevention of the spread of infection. So here is a list of some of the biological activities of interferon, and which will explain what they do and how they work. So what they do are these protein products that bind to cell surfaces and induce genetic expression changes. And the exact results are going to vary depending on the interferon that we're referring to and the cell that it is binding to. But all three of the various types of interferons that we've described, alpha, beta, and gamma, can inhibit the expression of cancer genes. It can have tumor suppressor effects. They can stimulate phagocytosis. They can act as immune regulators on various cell types. Those are just a couple of the activities that can happen when a cell interacts with an interferon protein. Moreover, these interferons tend to bind to viruses and other microbes. It binds via receptor sites on our host cells and signals the cells to produce products such as interferon. So when we have the binding, and I'll draw a cell here. Recall that they have receptor sites, and here's my poorly drawn receptor site. If we have some kind of microbe in the environment that has a receptor site on it that interacts with this receptor, okay, sorry, my arrow looks horrible, I apologize, but again, we have this receptor site on our cell. We'll say this is a neutrophil, hypothetically, a white blood cell, and it interacts with this protein on our bacteria cell. It can cause it to produce the cell interferon. And interferon is a molecule that's going to be then secreted in the extracellular matrix, and it's going to bind to other host cells. When I say host cells, I'm talking about your cells. So that's kind of cool because it kind of, in a way, introduces these proteins to all the cells in the surrounding area so that pathogen, whether it be a virus or a bacteria, etc., can't associate itself with other cells. So in the case of viruses, as we've talked about, what's their goal? Their goal is to get into cells. So if this cell recognizes this virus and starts pumping out all these interferon proteins, then this cell over here is going to recognize that interferon protein, and it's going to essentially be put on lockdown, and it's not going to allow anything else in the extracellular fluid, such as other viruses, to infiltrate it. So that's kind of cool. It inhibits entry of a virus. It can cause degradation of viral DNA, and it can also prevent translation of viral proteins, which ultimately is going to slow or eliminate the spread of an infection of a virus. These are not microbe specific. This can occur with any kind of microbe that has the capability of inflicting or triggering interferon production. So here we see a much better drawn picture than the one that I drew, but we see a viral infection occurring because this virus has now infiltrated this cell, and that cell has then began turning on its IFN gene, interferon gene. It starts making interferon. So on the other side here, we see the viral DNA is being copied and assembled, making new viruses, as per usual when we talked about that in chapter 5. But because of this secretion of interferon, which is now interacting with this nearby cell, it's told this cell, hey, there is a virus in the area. So wake up, do things. And it alters the activity of the genes of this cell, which will do various things, which can include blocking this virus from entry, degrading any of the viral nucleic acids that have entered, and blocking viral replication, which ultimately inhibits the spread of this virus in the body. Another example of some antimicrobial products that exist in our body is complement. Complement complements the immune system reactions. And it's very complex. We're not going to go into the details, but we'll talk about some things about it. It consists of like 30 some odd blood proteins that work in concert together to destroy bacteria and other pathogens. It has a cascade reaction, which again means it's a sequential process, goes in order like a domino effect, where the first substance in a chemical series is going to activate the next, which activates the next, and so on. Just like if you were to have a row of dominoes and flick the first one, it's going to cause a series of reactions to occur that end eventually. So our 4 stages of our complement cascade are as follows: initiation, activation, polymerization, and membrane attack. I do not expect you to know all of these details verbatim for the exam, but I'm going to tell you some of them anyway. In initiation, we're going to have a protein we call C3 that is going to be hydrolyzed, which means broken down into fragments that we call C3A and B. Then that C3B protein will cleave another protein called C5 into C5A and B. The C5B fragment is now going to form a complex with C6, C7, and 8. And collectively, they are now called the membrane attack complex, or sometimes that is referred to the MAC, and I like to call it the MAC attack because that sounds cute, I guess. But again, I don't expect you to know that domino effect, but know that the result of the complement cascade can lead to the membrane attack complex. That's the part I want you to know, the MAC attack. And so what this membrane attack complex is these various protein fragments that are going to infiltrate the plasma membrane of a foreign cell and cause it to form pores. And these little pores are going to cause water and basically any other excess material inside or outside of the cell to infiltrate, and it's going to eventually make the cells rupture. There are also some alternative pathways that the complement can take called the classical and alternative pathways. The classical pathway is initiated by binding to antibodies that are already bound to microbe, which kind of connects to our adaptive system that we'll talk about later in the following week. And our alternative complement pathway does not require antibodies to get started. It's quicker than the classical pathway and is actually instead initiated by the presence of the foreign cells' antigens themselves. So again, but my main concern, or the main thing I think I want y'all to know about it is that complement is a series of a bunch of proteins that have a cascade of events that leads to a response. And in some cases, this response is the MAC attack. And that's where we have these various proteins that are going to be that are going to work together, we'll say. For example, C3 here, you can see it is hydrolyzed, broken down to C3A and B. C3B binds to C5. That causes C5 to break down into C5A and B. And then we have C5B that will bind to C6, 7, and 8, I believe it was, to form the MAC attack. And so some of you, especially my biology students, I hope you're thinking biological pathway. Remember metabolic pathways that we saw in biology class? This is an example that we see in the immune system. But ultimately, this membrane attack complex boop, boop, together, now has formed a pore or a hole in the cell, which will result in lysis or rupture of it. Alright, furthermore, there are other antimicrobial peptides. Peptides are short proteins, typically about 15 to 20, up towards into 50 amino acids that are going to have some kind of antimicrobial property. One group of them is called defensins, which is kind of nice because it has the word defense in there. But what they do is they insert themselves into bacterial membranes and are kind of going to serve a similar effect as the Mac attack, where it will create a pore in the membrane, and if it's big enough or if there's enough of them, it will cause the cell to burst. And this is also a non-specific example of an action on a pathogen. Here we see in this picture an example of these antimicrobial peptides. As you can see, it's forming a pore. More specifically, how it works is there are certain areas on this protein that are hydrophobic and certain areas that are positively charged. And if y'all recall from biology or A&P, when you look at the cell membrane, these portions, the balls, are representing the heads of each phospholipid, and they are going to have a phosphate group in them, and that phosphate group's negatively charged. So what's attracted to negative charges? Positive charges. So that's why we see this section of this amino acid chain attracted to the phosphate group heads here and here. Now, the hydrophobic portion is going to push itself into the plasma membrane's insides. Why? Because these tails are lipids. They're fatty acids. And as you all should be familiar with, those are considered hydrophobic, meaning they don't like water. And what's inside and outside of the cell? Mostly water. So this hydrophobic portion of this peptide is going to force itself into the inside of the plasma membrane, and that's going to allow potentially fluid to influx through this passageway that it's creating and ultimately rupture the cell. Alright, and that is the end of this chapter, you guys. Here's another concept check question that you can utilize as practice to see how you feel as we move through this chapter, and as practice for a potential test exam question.